Hello, everyone. Thank you for joining us today for the first webinar in our EcoNAM series on the use of new approach methodologies in ecotoxicity. I'm Christopher Fassbender. You can call me Chris. I'm an advisor to PETA Science Consortium International, and I'll be moderating today's webinar along with Dr. Wesley Hunter, a supervisory environmental toxicologist with the US FDA. My colleague Tess Rienehan is here to help with any technical issues. Please feel free to contact her via the email on the screen. Before we hear an introduction from Wesley Hunter, note that the recording from today's webinar will be posted online. Please visit our website on the screen for the recording and more information on future EcoNAMS webinars. Wesley, please go ahead.
Thank you, Chris. All right, well, welcome to the EcoNAMS webinar series. As Chris mentioned, my name is Wesley Hunter. I'm an ecotoxicologist and chief of the environmental branch at the United States Food and Drug Administration's Center for Veterinary Medicine. And it is my pleasure to welcome you to this new webinar series that will focus on new approach methodologies in ecotoxicology. And on behalf of the organizing committee, we thank you for joining us. This webinar series was organized by several stakeholders, including regulatory authorities, research organizations, non-government organizations from North America, Europe, and Japan. And the organizers are listed on this slide and include the European Medicines Agency, the Health and Environmental Sciences Institute, the National Institute for Environmental Studies in Japan, the PETA Science Consortium International, the United States Environmental Protection Agency, and the United States Food and Drug Administration. The purpose of this webinar series is to share knowledge of new approach methodologies for ecotoxicology that allow for the reduction, refinement, and replacement of animal toxicity tests for ecological safety assessments. It is our objective to increase awareness of existing NAMs across stakeholders and how NAMs may be applied to environmental assessments. This includes tools that are already available as well as new tools that are in development. In addition to presenting information, we hope that these webinars will allow for interactive discussion. We acknowledge that there are different definitions of NAMs in the scientific and regulatory communities. In the context of this webinar series, we refer to NAMs as technologies, methodologies, or approaches that can be used to replace, reduce, or refine animal toxicity testing and allow for more rapid or effective prioritization or assessment of chemicals. One of the purposes of NAMs is that they may provide information of equivalent or better scientific quality than the use of vertebrate animals. For the purposes of this webinar series, NAMs may be categorized as shown in the figure on this slide. These include use of in silico approaches, in vitro assays with biological molecules or cells, the use of non-protected life stages or use of invertebrates, and the use of exposure estimates like threshold estimates or refinement of exposure dosing levels. The format of the webinars will be as follows. Each webinar will be 1 hour in length. This will include 40 minutes of presentation time, which will consist of 2 presentations of about 20 minutes each on the same overarching topic. The talks will aim to cover background information, description of the method or tool, and options for integration and application in environmental safety assessments. After the presentations, there will be time for discussion. Please note that the webinars, as Chris mentioned, will be recorded and published for future viewing on the YouTube channel of the PETA Science Consortium International. The topic of today's kickoff webinar is bioaccumulation. The speakers are Dr. Pippa Curtis-Jackson from the UK Environment Agency and Dr. Michelle Embry from the Health and Environmental Sciences Institute. Subsequent webinars will be held on a quarterly basis, and the topics and dates for future webinars will be announced in due course. Future webinar topics may include predicting acute and chronic fish toxicity, waiving fish in avian testing, validation of NAMs, application of 3Rs in pharmaceutical assessment, and NAMs for assessing endocrine disruption. To register for future webinars, please access the EcoNAMs page on the website of the PETA Science Consortium International. And before we start, I want to mention a few tips and ground rules to keep the webinar efficient and effective. You are invited to submit written comments or questions during the presentations via the Q&A function. If you cannot access the Q&A function, please email your comments and questions to tess@thepsci.eu. Please take note of the email now on the screen in the event that you do not have access to the Q&A function. After each presentation, there will be an opportunity for brief questions for clarification. When both speakers have finished their presentations and clarifying questions have been addressed, we will address additional questions for both talks submitted to either the Q&A or to Tess's email. In your written questions, please identify the presenter for whom the question is intended. And dependent on the number of submitted questions, we may later also consider inviting oral questions. In that case, we will invite you to raise your hand and we will unmute you when it is your turn to ask a question. And with that, I'm going to turn the time back to Chris Fossbender with the PETA Science Consortium International, who will introduce the first speaker.
Thank you for that nice introduction, Wesley. Yeah, now we come to our first presentation on bioaccumulation. Pippa, can you please show the first slide of your presentation? So Dr. Pippa Curtis-Jackson is a senior specialist in the Chemicals Assessment Unit of the Chief Scientists Group in the UK Environment Agency. She leads a sub-team of scientists who focus on the environmental hazards of suspected persistent, bioaccumulative, and toxic, and/or very persistent and very bioaccumulative substances. Dr. Curtis-Jackson is an analytical chemist by training, and she built her expertise in the specialist subject area of environmental fate and behavior in private industry. Thank you for joining us today, Pippa. The floor is yours.
Thank you. Sorry, I was unsure whether I had unmuted myself there. So yes, thank you for the introduction, and thank you very, very much for inviting me to speak today. This is a subject that I love talking about, as a lot of my colleagues will tell you. So yes, today I'm going to talk to you about regulatory and industry acceptance of new approach methodologies in bioaccumulation assessment. I'm going to give you a very quick introduction to the hazard and how it evolved. A very, very brief overview of bioaccumulation assessment, as I'm sure many of you know exactly what it incorporates. And then I'd like to move on to why we need NAMs for bioaccumulation assessment. And then finally, how we're going to overcome some of the challenges. And then I'm going to introduce some of the research and initiatives that we are currently involved in. So, okay, so chemicals. So from natural to synthetic, chemicals are everywhere, and the volume and variety of chemicals supplied has increased actually exponentially in the last 50 years. So it's really important to remember that chemicals are integral to our products that we use every day, and from fertilizers and pesticides that support our agriculture to coagulants, flocculants and disinfectants that keep our water clean, and medicines that ultimately help prevent and cure disease. However, although new chemicals are developed for their desirable functions, it doesn't mean that they cannot also bring problems. And many years ago, scientists actually recognized that there were properties of certain chemicals that really should give us a cause for concern, and these are your PBTs. And it's really important to note that when we're actually doing assessment on this, this is an iterative process. The logic here being if something is persistent, it's going to hang around, it's got a higher chance of actually being uptaken and accumulating in an organism, and that accumulation can actually lead to toxic effects being exerted. In some jurisdictions, we also have VPVB. This is mainly within Europe, but also K-REACH and certain other regulatory authorities. And this is based on the precautionary principle. And this is due to the fact that the long-term effects of these chemicals with these properties cannot be tested for toxicity in a laboratory setting. And then it purely becomes a precautionary conclusion. It's considered the better safe than sorry approach, and in contrast to our normal, let's just wait and see. So the protection goal of PBT is actually to safeguard ecosystems and human health. And they pose a unique risk because, as I said, they can contaminate a food chain, they accumulate in top predators, and it's the exertion of these long-term unpredictable toxic effects that actually we really would like to prevent. And therefore we try to prevent it by managing them. So where did persistence bioaccumulation toxicity start? Well, actually, these have been recognised hazards for a very long time. And back in the 1960s, there was a great rise in environmental awareness, but it was actually the 1970s that was the turning point in global environmental governance. And a number of regional and international agreements have actually been signed to tackle pollution and protect nature from substances with these chemical properties. And one of the earliest and most significant for PBT assessment was the Great Lakes Water Quality Agreement in 1972. And that was actually in direct response to a set of environmental disasters that actually included the Rouge River Fire in 1969, which marked a commitment between the US and Canada to restore and protect the Great Lakes. Building on this, the Great Lakes Binational Strategy was introduced in 1997, and it set a very bold goal in order to actually virtually eliminate human-made PBT substances from the basin. In 1976, the Toxic Substance Control Act was actually passed in the US and began to incorporate the PBT assessment guidelines into the late 1990s. And this was actually the basis for the Environmental Protection Agency being established in the US. On the international stage, the Stockholm Convention was adopted in 2001, and it targeted persistent organic pollutants based on their PBT properties, aiming to eliminate and restrict their production and use. That same year, OSPAR, which is the Oslo-Paris Convention, actually adopted it as well. And then it's important to note that all thresholds and data requirements and risk assessment approaches actually still vary very widely across jurisdictions. However, the PBT criteria we actually use today, and especially the screening criteria, were developed in the early 2000s, and these were inspired by POPs. In Europe, the criteria were revisited to capture chemicals that biomagnify in terrestrial food webs back in 2011. But even now, guidance for assessing bioaccumulation in air-breathing organisms like mammals is actually really only just become available. We still lack standardized methods for identifying substances that accumulate in non-lipid-rich tissues. So, what is bioaccumulation? So it's a gradual increase in the concentration of a chemical in an organism's body, and it occurs when the intake of a chemical happens faster than an organism can get rid of it. And these figures show that if we're trying to predict whether a chemical will bioaccumulate, we have to consider multiple exposure pathways. For example, what an organism eats, how it respires, whether it lives in water or air, and output pathways, which is what happens inside the body. Will the organism grow? Are we going to see growth dilution? Or does the organism have the ability to biotransform a chemical internally to something else which will allow it to be excreted, or is that going to encourage movement and distribution? And ultimately, how much of this is actually naturally removed by an organism through excretion? And all of these have different rates of uptake and depuration or removal, and this can vary depending on species and trophic level. Ultimately, we also need to understand where a chemical goes within an organism and what is it that's actually retaining the substance. Does it accumulate in lipid-rich tissues or is it protein-rich tissues, which of course all vary proportionally between organisms. If we try and extrapolate all of this to the wild, we get further variability, and that is because we have geographical and seasonal effects, and we also have multiple variables to account for, such as food and migration. To summarise, it's actually quite a complicated picture. So how do we currently assess it? So I work mainly on industrial and consumer chemicals, and we have a set of criteria which are set out legally in Annex 13 of our legislation. So, for example, first thing we would normally see is an optimal water coefficient or an optimal air partition coefficient, which can both be used in junction to either screen for aquatic uptake or uptake in air-breathing organisms. So these were derived historically because PBT substances are mainly associated with accumulation in lipid-rich tissues. And optimal is actually now the standardized accepted model surrogate for these tissues. And if a substance moves from water or air into optimal, it's then considered that this could be a way of passing into the lipid-rich tissues of an organism. If, when we're following screening, we think that a substance has actually passed the thresholds for being potentially B, then we'd normally need to go on to higher tier data. And our legal threshold is defined as a bioaccumulation factor greater than 2,000 or or very bioaccumulative, greater than 5,000. This is only for aquatic organisms. So we have a definitive test, and it's a standardized OECD test guideline, the 305, which allows for both aquatic and for dietary uptake. And both tests can actually produce a fish BCF that can be read directly back to a threshold. With the dietary study, we actually produce a biomagnification factor, and we actually apply a series of algorithms to actually read it back to a BCF. To add to our complications, especially within our area, industrial and consumer chemicals only have testing available, and it's dictated by the yearly manufacturing tonnage. So for very low tonnages, we see very little data. For very high tonnages, we will see much more data, and we may also see a bioaccumulation study, a definitive bioaccumulation study, being part of a registration requirement. However, we also need to bear in mind that many substances aren't actually suitable for actually being applied into these studies. So we also see a lot of data waiving, or we see a lot of modelling, and some of it isn't that well understood. This always appears to be a very simple assessment when we present it like this, but actually it's a huge weight of evidence and a process combining multiple lines of evidence. And actually Michelle is going to talk a little bit more about how these are evaluated and weighted in her presentation. So what's the weight of evidence approach? Well, beyond those screening and definitive criteria, bioaccumulation assessment is based on multiple lines of individual evidence. If we identify gaps that will not allow us to conclusively say something is not bioaccumulative, we can examine many, many more avenues. And this includes mammalian TK or ADME data. And those are the data that look specifically at input and output pathways. And where does the substance go, and how do they behave once they get into the organism? We also use field monitoring data, non-standard studies, and anything else that is considered relevant. The important thing is, once we've combined everything, we can read back or extrapolate back to the legal threshold and demonstrate an equivalent level of concern to a BCF, even if we haven't got that study with that value or data output. So why do we need NAMs for bioaccumulation assessment? Well, we've had some advances in understanding that have been incrementally incorporated into regulatory guidance, but there is a general need to revisit the fundamental science to allow for further improvements. We have to remember that both the screening and definitive criteria were developed using the best science available between the 1970s and the 1990s on chemicals that were known to be highly hazardous, and such as the POPs. The majority of them are neutral lipophilic organic compounds, and they accumulate normally within the fatty tissues, and therefore the oxonol water is fine, and we can probably assess that this substance may and actually screen in very well. However, the chemical universe has actually developed hugely in the last 50 years, and it is now highly complex, and it's always evolving. And many chemicals that are present are not actually these neutral lipophilic organic chemicals any longer, and don't fit the applicability domains of those established tests, and consequently they've been found to accumulate in alternative tissues. So beyond these issues are the practical issues of data availability. As requirements vary depending on registration tonnage, as I also mentioned, and we often lack data to really understand a chemical, and this means that reliance on the older models, such as the OECD 305 and data from the OECD 305, are actually not fit for purpose any longer. There's a clear need to improve the way we assess bioaccumulation, and therefore it begs the question whether we can use NAMs to fill these data gaps. So there's definitely an appetite. There's definitely an appetite to develop these NAMs for bioaccumulation assessment. The four topics that we routinely see when discussion, especially at a policy and a regulatory level, are acceptance through regulatory and organisational barriers, validation and standardisation, biological complexity, which is, when we're extrapolating from an in vitro cell line to a whole body organism, how do we know we've done it properly? And finally, species diversities. How do we overcome the fact that we are trying to protect the entire wildlife and subsequently humans? So can a NAM therefore replace an established in vivo study? For most substances, actually, this should be possible, but we've got to be mindful that UVCBs and complex reaction mixtures and certain unusual chemistries that are very difficult to test may not be so easy to adapt. And therefore, that is where our models need to come in, and that's our in silico models need to get stronger. We also need to be attentive that a NAM approach, which was originally developed from next generational risk assessment and human health tox, considers exposure and mechanistic information using a range of in vitro methods, data and computational models. And this doesn't easily align with a hazard-based regulation, such as REACH or UNGHS, where the paradigm relies on identifying and characterizing hazards using internationally harmonized guidelines. However, there have been many advancements in the bioaccumulation science since the principal criteria were developed, and we're drawing on these advancements in our projects to see if we can actually build this to being accepted as part of an internationally accepted or validated method. And I'm going to talk you through some of those research projects now. So one of the major areas we're supporting are improvements in our understanding of the influence of toxicokinetics, specifically biopartitioning and biotransformation. And this work was initiated following several substance assessments that revealed limitations in using the optional water partition coefficient as a screening tool. These assessments also noted unusual tissue distribution patterns, which differed significantly from, as I said before, these NLOCs, this neutral lipophilic organic substances, upon which most of our understanding is based. And subsequently, we came up with a project which was titled with this very short and snappy title of Scoping the Uncertainty in Screening for Bioaccumulation Potential of Substances for which Optimal is not a reasonable simple surrogate for partitioning, including an assessment of alternative surrogates and toxicokinetic methods. You will be glad to hear it is no longer titled that, and we now have 2 reports underway. So the first phase of this work focused on identifying alternatives to octanol traditionally used. And we asked the question, can we find a surrogate that presents potentially proteins, phospholipids, and other tissues? So starting with the proteins, 4 main types of protein were identified, which were soluble, membrane, and that includes your globular proteins, fibrous, and disordered. And chemical affinity to these generally falls into 2 categories. So we either see specific binding, much like an enzyme fitting into a receptor site to trigger a response, or non-specific partitioning, which is actually the dominant passage into the tissues for most substances. So the review found that globular proteins are the most representative for whole body partitioning and bioaccumulation potential. It identified some commercially available proteins suitable for lab-based screening tests, and galactins and carbonic anhydrases are actually proved to be the most promising, even though carbonic anhydrase is an enzyme and involves specific binding. The phospholipids, which of course is the formation of your cell membrane with the hydrophilic head, hydrophobic tail, and we would like to further understand chemical accumulation in these structures, and we actually think this is quite crucial. The review also identified unilamellar liposomes, which actually offered some flexibility in experimental design, but it's proven that actually it is very labour intensive, and immobilised artificial membranes and solid supported lipid membranes, which require more setup and care, could enable higher throughput testing. So this foundational work has set the stage for us actually going into the laboratory with a little bit more work to do, and that's going to be undertaken in the next year after the finalisation of the report. So the second part of this project actually went into toxicokinetic modelling. So TQK models allow us to consider all potential input and output pathways, such as uptake, distribution, metabolism, and excretion, and how they influence bioaccumulation. A refresher of these pathways is in the top right-hand corner of the screen. So we reviewed a lot of existing models that are built on the previous assumptions with the octanol. We compared BCF data on the y-axis and the predicted values on the y-axis. And the data actually showed that the bioaccumulation estimation tool, which is part of the Easy Suite platform, performs significantly better in this comparison than other established models. And this can be seen because we've got a nice one-to-one line indicating higher accuracy with the outputs. So once we established the use of the BEC, we explored its capabilities in several other ways. I'm just going to highlight a couple of findings here because these are the ones that we think are actually very, very interesting. So we assessed how well the tools predicted biomagnification factors, which are, of course, very important when we're looking at food chains. And we actually reduced this to the air-breathing organisms. So the graph on the left shows a good level of agreement between predicted and observed values. But in general, when a substance known to biomagnify in the field, and it's actually shown that we can see that substance is actually, there's a very good one-to-one with substances that are known to biomagnify in the field, and the model is actually categorizing this correctly. And then we actually had a look at the impact of including internal biotransformation. And this was included in the BMF predictions. And the graph on the right compares the predictions with and without biotransformation. And the red line marks a BMF threshold of 1. And as you can see, when biotransformation is actually included, substances are actually generally predicted not to biomagnify. But when biotransformation is excluded, the same substances are predicted to biomagnify. And therefore, the chart on the right is actually showing a better correlation with what is actually known to biomagnify in field studies. So it shows how critical it is to accurately characterize a biotransformation in an assessment. And the key takeaway from this phase of the project is that whilst the BET tool is actually already useful for screening, there are still data gaps and it needs to be addressed to improve its accuracy. And although understanding in biopartitioning is important, it's even more crucial to get the biotransformation right. And because of these findings, this project is actually being extended into this year and next year, with the work imminently due to start. And this is going to include collation and integration of biopartitioning data for proteins, and their database is going to be compiled and it's actually going to be integrated straight into the Easy Suite platform, which is freely available. So hopefully other people will get to play with it as well. We're going to have the parameters will be expanded and recalibrated, and then we're going to run predictions for around 80,000 substances, replacing the optional surrogate assumption. We're going to be performing a few case studies as well, and hopefully this will allow us to enable the impact of bringing in vitro data from the OECD 319 model. We're going to be doing further in silico model validation, and we're going to be looking at the impact of the on elimination of half-life in fish for around 20,000 substances as well. So the second project is our work that we've been looking into with aquatic invertebrates. So many of you know that the HYBIT was developed, it's now an accepted test guideline at the OECD. So I won't go too much into that, but we are also very interested in Daphnia because we believe it offers several advantages over the Hyalella. So we commissioned a review back in 2023, which is based on the evidence review, collated as much data as we could for Daphnia and bioaccumulation toxicity studies. We were looking at body burden, and we identified that there are a lot of uncertainties when you are looking at aquatic invertebrates to do an uptake. So some of this is now being investigated in the laboratory. We're fairly confident that it's going to come out with quite positively. And ultimately, what we're going to be trying to do is adapt the OECD 202 and the OECD 221, the Daphnia acute and chronic, to actually be able to run a bioaccumulation study in tandem with a tox study, which is a data requirement for most chemicals. And the laboratory work for this is starting in October. So some good news. Another piece of work that we are also involved with, Michelle is going to talk about this as well, is the OECD update of the guidance document for the 280. So back in 2018, the OECD endorsed the NAMs for bioaccumulation assessment for test guidelines 319A and 319B. So these tests are fish liver-derived cells, got significant animal welfare benefits. They're much faster to perform and about 75% cost reduction compared to doing a 305. So the accompanying GD 280, it's actually the foundational guidance for actually applying these data as a line of evidence. And back in May of this year, we, HESI corralled an international multi-sector project team, which includes the test guideline developers and the IATA bioaccumulation authors and other experts, in order to discuss how we can improve and update the guidance document. And especially the focus of this will be, we're going to be updating against current science and understanding, which will include test selection and methodological advances that have happened in the last few years. We're going to be updating the in vivo models, which are being ground truthed with in vivo BCF data, sensitivity analysis is being performed, etc. And this is a huge part of the exercise because everything is being done very controlled and very validated to hopefully improve regulatory confidence in it. Broader applications being considered also include species extrapolation and metabolite AOD as well, which is always very handy. Finally, just wanted to touch on the BIATA. So this is Michelle's going to talk you through this in the next presentation. It's really, really important project that we were involved with. So we absolutely really do think that being able to bring everything together consistently and transparently in a weight of evidence with the assessment is so important, and actually very important for the NAMs as well, because this method is very, very robust. So what next? So for us, we're going to be finalising the biopartitioning and biotransformation work. So hopefully next year, commissioning further work based on this. We've got the empirical data coming through from the Daphnia screening studies, and hopefully in a few years' time, we'll talk about development or amendment to existing test guidelines. And the UK OECD test guideline coordinator, we're working closely with her with the submission for the update to the GD 280. So final thoughts. Okay, so this is very much my opinion. So this is not the opinion of the organisation I work for necessarily. So to confidently move to alternative methods, I think we've got many areas of this complex subject where further understanding is needed, and the tools need to be improved and updated. We need to be leveraging other knowledge from other sectors, and we need to find a way of doing this relatively quickly and cheaply. We've got an increase in substance complexity, and I think this means that we need to go back to the first principles and the protection goals, and we can't do this without examining the foundations that our current understanding is built on. And one of the things I would like to see if we could encourage to be done sooner rather than later is a full update of our in silico tools. And this could involve collation of all data from 2009 to today. We could then expand and recalibrate a lot of our established estimation models. We could improve the chemical space and applicability domains. This would allow us to reduce uncertainty in the outputs and improve regulatory confidence. And finally, I probably had enough of me talking now, but I'd say a huge thank you to my team. So that's Steve, Richard and Steph here. Huge thank you to our sponsors at DEFRA who actually paid for this research funding and the work. Michelle, a very valued colleague, who I enjoy working with hugely, the BIATA team and the team at Arno Research and Consulting, and to the HESI bioaccumulation team as well. So thank you very much. Any questions?
Thank you, Pippa, for that great presentation. If any clarifying questions have been submitted, they can be briefly addressed now. Any other questions will be answered after the second presentation. But the questions we have so far are not clarifying, I'd say, so let's discuss them later. And therefore, Tess, do you have an announcement?
We have one clarifying question. Chris, do you see the Q&A and in the document?
Okay, yeah, then let's look at that question. Thank you for this presentation. Do we have a comparison on the results provided by the biopartitioning screening test, surrogate screening test, compared to the optimal partition coefficient and the final BCF?
It will be available when we've reported. So this will be next year. So that all the data will be there, and the raw data will be included in the report.
Okay, thank you so much. It looks like we don't have any other questions for clarification at the moment.
I think there was one, Chris, on the Daphnia model. Do you see that one? I can just read it.
Wait a second.
It says, can you clarify the objectives with the Daphnia model for me? Do you plan to add this model to the 321 test guideline or to the 202 and 211? Or do you plan to publish a new guideline combining ecotoxicity and bioaccumulation with this invertebrate organism?
That's a very good question. So what we've been doing, we've really based it on the 201, the Daphnia tox studies, and the idea was to add an additional tool at toxicity screening when we initiated the project. It's unlikely that we would develop a new standalone test guideline because ultimately, I mean, one of our long-term goals is to start understanding body burden in terms of toxicity as well. So we combine bioaccumulation and the T together. And if we are running essentially the different endpoints together in each batch, we think that we could actually achieve this much more quickly. It would be an optional. It would be added as an annex, hopefully, to a test guideline in order for this to happen. It's still early days. We still have a lot of work to do, and the empirical data is really going to guide us with where we go with it. Thank you.
Okay, thank you so much. Please submit your further questions anytime, and you can submit them via the Q&A or via test via email. And note whether the questions are for a specific speaker. We move on to our second presentation on bioaccumulation. Michelle, can you please show the first slide?
Yes, can you see it?
No. I can't see it yet. Can anybody see it?
No, and Michelle looks frozen to me.
Oh, yeah.
But there are some more questions if we want to cover those for Pippa while Michelle reconnects.
Okay, let me refresh the Q&A.
I can go ahead and ask a clarifying question. Whether testing on BCF is required for generic application or data from Innovator can be referred.
Sorry, could you repeat the question, Tess?
Can you please clarify whether testing on BCF is required for generic application or data from Innovator can be referred?
For industrial and consumer chemicals, it depends on the tonnage, and it's being able to either justify why you're going to use a different metric if it's actually you are meeting the requirement for a bioaccumulation study, and if your substance is suitable for testing, you also need to look at that very carefully because it can be waived, but there will be other pieces of data may be needed. Does that answer the question?
Seems so. Then let's ask the next question and hope that Michelle will rejoin. How many definites regarding biomass would you need for determining the internal concentrations?
That is a very good question. It was one of the things we've been trying to answer. However, we do know there is actually work that's been published. I'll put the papers in the chat later. So there is work that was published that was done between the University of Vienna and the Berlin Technical Institute, where I believe they were taking 30 per replicate. There's also work that's going on at the University of Brunel as well. They've been developing a high throughput screening method, which is quite exciting. And I believe they were around, let's say, between 20 to 30 definites. If you want me to give you a link to the Environment Agency report, you can see how many different tests there were where we could see the variation in the actual mass needed to do a full body burden assessment. I can pop that in the chat.
I don't think people will have access to the chat.
I think we have Michelle back as well.
Yeah, I don't know what happened, but Tess, do you have my slides if you could share, if that's possible?
Yes, please give me a moment. And Chris, can you continue with some of the questions listed?
Yes, apologies.
Yeah, let's do another one and then continue with Michelle's presentation for the sake of time. Pippa, can you expand briefly on your thoughts between the Hyalella and Daphnia magna bioaccumulation methods?
So the methods are, I mean, they are going to look very, very similar. And depending on your test substance, it will depend on how you do a static or a semi-static, and with a Daphnia, we would never do a flow-through study with them. I think our concerns with the high bit, it's the same with the Daphnia. There's the presence of food in the test system, because that gives you a BAF, not a BCF. With the Daphnids, we're trying to answer the question about it's okay not to feed them with it for a 48-hour period, but it's not okay longer than that. You actually start to impact their health. Daphnids are only one sex, so we only have females, whereas the Hyalella, there's a lot of work goes into actually removing the males and females from each other. And generally, there's actually a lot more data for Daphnids. They're so well studied in terms of genomics, etc. And there's work going on at the University of Birmingham looking at it at a toxico, the actual genetic changes with toxicity. So there's a lot of threads to pull here, and we just felt that the Daphnia, just because it's been so well established, was actually going to provide slightly more robust data, and that would be accepted because the organism is actually very well studied.
Thank you, Pippa. For the sake of time now, let's move on to Michelle's presentation. Our next speaker is Dr. Michelle Embry. She is the Deputy Director of the Health and Environmental Sciences Institute, a non-profit organization with a mission to develop science for a safer, more sustainable world. Dr. Embry holds a PhD in toxicology and a bachelor's degree from Duke University. She has 20 years of expertise in human health and ecological risk assessment, with a focus on methods and approaches to ensure safe and effective use of chemicals and products. Thank you for joining us today, Michelle. Please go ahead, but I still can't see your slides, unfortunately.
Okay, I'm sharing again here.
Yes, there we go.
Okay. I apologize for can you hear me okay?
Yeah.
Okay,
very good.
Great. I apologize for the technical difficulties. Microsoft decided it wanted to update right when I was presenting. So I know we're a little short on time, but I appreciate the opportunity. I'm thrilled for HESI Global to be part of the EcoNAMS webinar series. And as Pippa mentioned, I will be giving an overview of an IATA that was recently developed for bioaccumulation assessment. Pippa did a great job of giving you an overview of bioaccumulation as a regulatory requirement and the methods available and some of the research that's happening. And I will be really focusing specifically on this IATA and the whole purpose of this, which is trying to integrate different lines of evidence together for bioaccumulation assessment. So I have to give credit to my co-authors. This IATA was developed over a period of about 2 years or so with the team listed there. And we all worked together as a team to pull all this information together. So this was actually, it's listed as 2023, but it actually came out under the OECD IATA program in 2024. I think it was November of last year when it was officially published. And a lot of the purpose of why this team came together was to try to provide a little bit more structure and clarity because there are so many different pieces of information that go into a bioaccumulation assessment. And I'll walk through that figure there in a minute, but this is just a really high level overview of the IATA, and the link to it is provided and my slides will be made available down below, and I just encourage you to go and check that out and read more about it. So why did we decide to do an IATA? Pippa covered this, but the OECD IATA program, or integrated approaches to testing and assessment, is really a way to bring together the diversity of knowledge to allow additional clarity and consistency. And as Pippa covered already, there's a lot of different metrics that are used to help us understand bioaccumulation of a chemical. There's also various regulatory programs that have different criteria that go into this. And of course, as was already covered, there's resource limitations and a movement towards integrating non-animal methods and new methods and alternatives into this assessment. And of course, with any method, there's inherent uncertainty in both measured and predicted data. And there really is a need, and I think over the last 15 years or so, there's been a real increase in the metrics that are available to evaluate bioaccumulation. We sometimes call it a bit of an alphabet soup over, all the different types of ways that you can evaluate a chemical's bioaccumulation potential. So we really needed some sort of framework to bring this all together. So for those of you not familiar with the OECD IATA program, the link there is to that provides an overview of that OECD program, but it's really intended to be a formalized approach that can integrate and weight different relevant existing evidence. And this can be everything from phys-chem property information, in silico models, in vitro methods, in vivo tests, as well as in some cases, field data that are collected for bioaccumulation. It can also guide targeted generation of new data when it's required, and I'll talk about that in a minute. And then a lot of it is really intended to combine sufficient information in an efficient way to help you get to definitive conclusions if needed, or to provide information on prioritization. And I would just point all of you to the IATA program. I think it's a really amazing program. It's not a test guideline program, but it's a really great opportunity to bring together different stakeholders that are working on state of the science to try to, especially in this current environment of a lot of NAMs and new information being generated, I think it can really provide a lot of structure and guidance. So this IATA did not just sort of spring from nowhere. It really was building on now decades of work in the bioaccumulation space, and much of it was building from the bioaccumulation assessment tool, which was another collaborative program led by Arnott Research and Consulting, but was really a multi-stakeholder government, academia, industry effort over several years to develop a tool to do this integration. And I won't go through all those bullet points, but if you can sort of see what I just talked about in the last slide, a lot of what this tool was aiming to do was to try to do what the IATA program is aimed at, which is to integrate, collect, allow transparent discussion of different lines of evidence. And this IATA leverages the foundation that was provided by this bioaccumulation assessment tool. So you've seen this figure already. This is the overall IATA workflow that I'll just walk you through briefly. It starts with the problem formulation, which is really that purpose of the assessment. And one of the things that Pippa talked about and I highlighted in one of the previous slides is there's different regulatory guidelines, there are different criteria, there's different overall purposes of the assessment. Are you looking at biomagnification? Are you looking at fish bioconcentration? Are you trying to look at sort of PBT criteria and how that fits in? Are you trying to look at how bioaccumulation can impact the larger scope of ecological risk assessment? So defining that problem is really important because, as we know, different lines of evidence are going to be fit for purpose depending on that problem context. So we spent a lot of time within the IATA talking about this and making sure that that was very well defined. The next step with all of this is moving towards evaluation of phys-chem property information, KOW, KOA, solubility, those types of things, and then moving down this workflow. One of the important things here that you can see is that these circles here are really meant to they're meant to signify the iterative nature of this. So at every step of the process, you would evaluate the information, assess the reliability, integrate it with other lines of evidence that you have, and ask the question, do I have sufficient confidence in the weight of evidence to make a decision? And if you do not, you can proceed to the next step in the workflow. If you do have sufficient confidence, you could stop or pivot to look at a different problem context, etc. So even though it looks extremely linear as conveyed here, it's really an iterative process, and you'll see that in the way that we pulled it together. One other thing that I just really like to highlight in this workflow is that we have biotransformation as a step past physical chemical properties. And Pippa talked about this a little bit with the 319 in vitro biotransformation data. We know that biotransformation is one of the key metrics that helps to influence and inform a chemical's propensity to bioaccumulate. So one thing that's a bit different is that within this IATA, we've really put biotransformation as a step before you even move towards sort of consideration of laboratory experiments. So I already covered this, but the problem formulation, really the point of this is to define the purpose and the context of the assessment. Is this for prioritization and screening? Is this for application of GHS? Or is this for substance registration or getting at specifics of the criteria, or maybe even the broader application into ecological risk assessment? And this is really important because it's critical to understand that problem formulation, because that gives you an idea of the acceptable level of uncertainty and helps to identify what your relevant lines of evidence are. So here's two example problem statements. The first one, is there sufficient evidence to conclude that particular substance you're evaluating meets a regulatory BCF and or BAF criteria of greater than or equal to a threshold? And that could be 5,000, it could be 2,000, depending on your regulatory jurisdiction. So that's one. A second one is maybe more field focused. Is there evidence to conclude that it has a biomagnification and or trophic magnification factor of greater than 1? So this could be used maybe for hazard assessment, monitoring, or compliance programs. And those two different problem statements would help to identify the relevance of the lines of evidence. So this is kind of the overall flow that I'm going to just be walking through with a simple case study in the pretty quickly here. One of the things that we tried to do within this IATA, in addition to explaining in great detail all the different lines of evidence, is to really get to, we have a lot of information, how do you integrate it? And that comes in sort of several steps. The first is defined by the problem formulation, the relevance of the lines of evidence, depending on that problem context. That information is combined with the reliability of the information, right? How robust is your study? What level of uncertainty do I have in that information? And those two pieces, the relevance and the reliability, come together to give you an overall weight for that particular line of evidence. And we didn't do a quantitative scoring, we sort of just have this high, moderate, low. That is a combination of the relevance and reliability. That information is then zipped together or integrated to give you an idea of your overall strength of evidence. And we've identified this within the IATA as a total percentage of the lines of evidence supporting various bioaccumulation criteria. NB, not bioaccumulative, B, bioaccumulative, or VB, very bioaccumulative. And the whole idea is to be able to use the outcomes to assign a weighting to the data so that you can look at that information and compare the strength of the evidence to the hypothesis or that problem context that you have. So all that comes together into three, we have the IATA identified, we have kind of the initial chapter that walks through the overall approaches, the information on lines of evidence, criteria, etc. And then the last part of it is 3 illustrative case studies that are listed here. They are illustrative and they're not intended to reflect any specific regulatory decisions or anything like that. We have 2 data poor case studies and 1 that's data rich, which is based on a publication that came out in IEAM a couple years ago. So I'm going to walk through one of our data poor case studies just to give you an idea of how this works in practice. So this chemical is a dicyclohexyl phthalate. The structure is there with the CAS number. And again, it's an illustrative example of a data poor case study. So our problem statement, and in the IATA, we actually go through 2 problem statements, but I'm just going to focus on the first sort of more simple one. Is there sufficient evidence to support the conclusion that this chemical, DCHP, is bioaccumulative in fish? So it's really looking at, could this chemical bioaccumulate relative to water using the bioaccumulation and bioaccumulative and very bioaccumulative criteria thresholds of 1,000 and 5,000. So we're looking at 2 different B criteria. So that's our problem statement. So we're kind of here in the yellow. And then we move down. So our next step really is trying to, based on that problem context, assign relevance scores. We kept this pretty simple here, with 0 being not relevant and 5 being extremely relevant. And what I really want you to focus on here is we have different lines of evidence that are all part of this IATA. And one thing I just want to mention is the IATA is meant to be flexible. So as new data come online, as new methods are developed, they can be integrated. This IATA is not prescriptive, saying that it only considers this type of test. It has that flexibility. So we have different lines of evidence in silico, in vivo, different lines of evidence and different bioaccumulation metrics, different organisms. And what we've done here is we've really identified in this case for this problem statement that our fish in silico have higher relevance. So our focus, of course, with the problem context is on fish, and we've weighted our in silico metrics lower than our in vivo metrics. And then we have our in vivo as a 5 in fish. Our invertebrates are given a 0 because our problem context was based on fish. And we aren't looking at field metrics for this because we're really looking just at BCFs. So I'm going to go through this pretty quickly. The next is phys-chem properties. Many of you are aware of KOW criteria. We have log KOWs greater than 4.5. So for most regulatory jurisdictions, that bumps us into having to look at this chemical more closely. So in this case, at the phys-chem property level, we have really great concordance across our different KOW measurements, and we proceed to the next step of the workflow. So moving on to biotransformation, in this case, we don't have any measured data for it, but we do have fish QSARs that give us values of whole body fish biotransformation rates. And one of the things that you can see here is that the 5 fish half-life biotransformation predictions are all very consistent. They're all in the domain of those fish KMQSARs. And we decided to use the consensus of the 4 modeled values of 14.2 hours to move forward to the next step. So that information, that biotransformation estimate, is used to inform the model predictions to kind of get us an idea of laboratory bioaccumulation. So that information is just a line of evidence. It's not giving us a specific yes, no, do I have enough information to stop, but that feeds into the next step. This next slide is really busy, but what I want to show you here, if you look at the white, these are our in silico bioconcentration values, and the yellow is the bioaccumulation factor measurements. Bottom line is that 5 out of 6 of the in silico predictions indicate that DCHP is not bioaccumulative in fish. It's below 1,000. 6 of the 7 BAF model predictions indicate it's not bioaccumulation, it's below 1,000. And then 2 of the 13 are above the threshold. So we have 2 that are a little bit that are higher than 1,000, 1 that's over 5,000. I just want to highlight that that high prediction is actually due to the fact that the EpiSuite regression doesn't include chemical specific values for biotransformation rates. And actually with this chemical, what we know is it's an ester, and it's actually very well known that these chemicals are rapidly biotransformed in fish as well as other animals as well. So we do have one line of evidence that's showing a VB prediction, but all the other ones are extremely consistent. So how do we look at this? All of these predictions, the lines of evidence, they gave us various B categorizations. They all had relatively high relevance, meaning they fit in the domain of the models. We don't have any in vivo or in vitro data for this chemical. Therefore, these were all highly relevant, had high reliability. So we weighted all of them very highly. And then we look at the information that we have. So one of the things that's great about this IATA is it allows you to transparently look at all the different lines of evidence together. We have our in silico lab BCFs and our in silico field BAFs sort of listed separately here. 85% of, again, the total lines of evidence that we have show that this chemical is not bioaccumulative. We have one, we're calling it an outlier from EpiSuite, showing that it's very bioaccumulative, and then we had one BAF prediction that was over the threshold of 1,000. So where do we go with this, right? So what this IATA does is allow you to transparently evaluate and demonstrate all of the information that you have. This is a very data poor chemical, and it's limited, the evaluation is limited to information from in silico approaches. But one of the things that I'd like to highlight with this is that despite the fact that we don't have empirical data, the in silico predictions are relevant, of high quality, and I think most importantly, very consistent across multiple lines of evidence. So we have multiple in silico predictions, and what you can do is look at those and say, are they consistent across? No further data for this chemical are available. One of the things we try to do with this IATA is we're not prescriptive. So you array the information in a transparent way. It allows you to have a dialogue with colleagues, with other regulatory agencies across sectors to discuss what this information shows you. Do we need to collect any additional information or not? And so I think one of the things that is somewhat unsatisfying is it's not prescriptive. It doesn't give you the magic answer at the end. But what's most important is it allows a very transparent array of the information for those dialogues to happen so that informed decisions can be made. Do we need some additional NAM data to be generated? Are there other chemicals that are similar that we can look at, et cetera? So it's a very informative process, and we're really excited to see other people try it. So just really briefly, there are a lot of things happening in the bioaccumulation space that Pippa highlighted and others. We would love to see people working to do additional case studies or to share with us their applications of this IATA. As Pippa mentioned, HESI and other partners were working to provide additional clarity on the IV-IVE model for fish biotransformation methods, both for neutrals and ionizable chemicals. That includes a revision of the guidance document 280. There's also quite a bit of work ongoing to collect existing in vitro fish biotransformation data. We have a subgroup that's looking at evaluation and integration and discussion of the recently adopted HIBIT method. And then I do also want to highlight that we've been focusing on fish, but there's some work ongoing. Marcus Brinkman's group at University of Saskatchewan is working on an avian in vitro biotransformation method. This work is funded by HESI, and it's very exciting to see that coming online. There's also a lot of outreach and coordination through the CTAC Bioaccumulation Interest Group, and I would just invite you guys to go and join that group if you are interested. They are doing a lot of work, and HESI and other partners are working closely with them. In the NAM space, this isn't bioaccumulation specific. There's a QR code there. We have a big meeting coming up in October in Reykjavik. This is really intended to bring the eco-NAM community together, and our first two themes are going to be on fish acute testing and endocrine testing. So just wanted to highlight that meeting. If you're interested, please feel free to email me. And with that, I'd just like to thank all my collaborators listed there, the EcoNAM group for involving us not only in this webinar, but also as a partner, and would encourage any of you, I know there's a lot of people on this call, that QR code will send me an email. We have a bioaccumulation group within HESI, a next generation eco risk group, and also I work with a lot of the other collaborators on this webinar series. So would love to hear from you and love to talk more about bioaccumulation and or this IATA if needed. Thank you very much.
Thank you, Michelle, for that informative presentation. With that, let's move to some clarifying questions. Can you please clarify whether testing on BCF is required for generic application or data from innovator can be referred?
I'm not sure. So and Pippa might be able to speak to this more, every regulatory jurisdiction has different requirements. So if you look at sort of many of them, the sort of baseline is looking at something like an octanol water partitioning, which might trigger the need for in vivo testing. But again, a lot of what we're trying to do and where the field is moving is to try to integrate other approaches other than, say, the standard FISH 305 test to allow sort of additional lines of evidence related to requirements. I don't know, Pippa, if you have anything else to add to that, but it really depends on the jurisdictions related to kind of what those requirements look like.
Yeah, absolutely. All data, every jurisdiction is slightly different. For example, Japan currently doesn't accept invertebrate data, whereas we would use it as a line of evidence. So yeah, please double check what your what the regulatory requirements are.
Okay,
And then I see another one about the water as the reference. So that really, the bioconcentration factor is looking at the accumulation of the chemical in an organism relative to the concentration in the water. That's a standard kind of evaluation for a bioconcentration factor. And then there's other ones that look at there's the bioaccumulation factor, the biomagnification factor, that are looking at concentrations relative to other environmental compartments. But the BCF is just a standard metric that's looking at concentration relative to the water.
Thank you.
I see another one on how these methods can be applied to degradation or biotransformation products. So there's a lot of interest in the in vitro method, the 319 method, to help identify biotransformation products, and then whether those could be of concern for bioconcentration, bioaccumulation. Obviously, what you have to do with all of these metrics is have that defined substance and be able to evaluate that sort of separately. So these methods could be applied. It's just a matter of identifying what those degradates and biotransformation products are, and then using that within the same models. The models themselves don't have, they don't have sort of an add-on that says, and also it's evaluating the products. It's a matter of the user identifying what those are and moving those through the process.
Okay, I think then, thank you. We can then move on to the questions that are not clarifying, and that would be addressed to both our speakers. Here I have one. Has there been any discussion in how to harmonize relevance scoring and reliability assignment to specific data to at least promote best practices to guide researchers?
Yes, I think I'll talk about the reliability. So I didn't cover this, but one of the things that's in the bioaccumulation assessment tool and also part of the IATA are data evaluation templates for each line of evidence. So for QSARs, you have like the QSAR model reporting framework format. For other metrics, we have sort of standardized evaluation spreadsheets that can be used to help with scoring. They have things like, all the questions that you would answer. It allows you to either use a numerical score, or you can do more of a qualitative evaluation. But the data evaluation templates in the bioaccumulation assessment tool and part of the IATA do provide numerical scoring for each line of evidence. They also provide a way to flag critical fails, for example, are there particular aspects of the test that would make that test just absolutely not suitable. Those go beyond, I think, the OECD test guideline discussions. And that's a really great question, because there's a lot of work that's been done on that. And I would just point you to look at the IATA to get more details on that. For relevance, that's sort of the beauty's in the eye of the beholder on that, right? So a lot of the discussions we had were, for example, certain regulatory jurisdictions would not put a high weight on invertebrate tests, for example. So some of that is really just trying to make sure that the user, as they're walking through, has defined what they feel like are the most relevant tests, or maybe those are defined by particular regulatory jurisdictions. But that's not prescriptive, and that's something that I think that what we've learned is, because harmonization across the globe for this is probably a nice idea, but something that's going to be much harder to operationalize. We left that somewhat flexible, but the reliability is absolutely codified in the IATA, and is something that we really feel strongly about.
Thank you so much. Wesley, would you like to pick one of the questions?
Yes. I think this question is from Michelle. How many in silico tools are required to be considered as reliable?
Ooh, that's a good question. I don't think there is a number. I think the key is to look at the tools that you have available, their relevance related to the chemical's domain of applicability. Those are the ones that are currently available for bioaccumulation, the ones that I showed for that chemical that are all within the domain. So there's tools within EpiSuite, there's tools within the EasySuite platform. So I don't think there's a magical number, but I would say for most of these in silico models that we have for bioaccumulation, we have multiple models available across different trophic levels within the existing tools that we have. I don't know, Pippa, if you have any thoughts on that. I don't think there isn't a magic number, but
there isn't at all.
The thing we wanted to highlight is looking at concordance across multiple predictions is stronger than just looking at a single prediction.
Absolutely, and one of the best ways for actually looking for concordance, especially where you're relying only maybe on screening level physicochemical data to build it in, is to actually look at the way the models work. They can either be built on different types of descriptor, they can be built on from fragments, they can be built on, iterative things, but it's the importance is the concordance around those values that are generated and how do they compare, because that can actually help reduce uncertainty.
Thank you. And I'll ask the next question. When we have in silico predictions from multiple in silico models, is it advisable to always consider a consensus approach, meaning taking an average value, or take the worst case if both minimum and maximum values can be defended or justified?
In principle, it's being able to defend your minimum and your maximum. I think especially screening and consensus, again, as I said, if you have got more of a closer your values are on each of the different types of models built from each of the different types of methods, your consensus value will actually be stronger. If you are basing your conclusion on essentially the worst case scenario, I would suggest you do a sensitivity analysis around that as well when you're doing the work, because you may have a worst case scenario because the substance actually doesn't fit the applicability domain, it doesn't fit certain other things. So, and this is why trying to strengthen the models with as many different chemistries and as much different forms of in vitro and in vivo data as well, it will just make everything better.
Yeah, I would underscore that. I think the key, if you do have multiple model estimates that diverge, right, you have a pretty large maximum and minimum, it's trying to then ask yourself, can you explain why there is that? Like I did with that EpiSuite one, right? And making sure that you really understand what your model is telling you before you just pick a maximum or a minimum or whatever that is, right? Understanding it and describing it, which is one of the things we talk about in the IATA, is making sure that there is that, again, you look at all the information, you array it in these tables, and you make it very easy to have that discussion and understanding.
Yeah, absolutely. Because you don't necessarily need, you can actually have fairly good confidence in your output. I would recommend having a look at, within the BIATA, there is a fantastic paper that's written by the team at ARC Research and Consulting, which is about how to address uncertainty in models. And the explanation there is fantastic. It was where I would recommend going.
I can ask the next question. This one is, we have, actually, I'm going to combine a lot. There are several questions about regulatory acceptance of NAMs. This might be maybe Pippa more focused towards you, but also Michelle, if you want to. Others are related to, although maybe the UK is advancing the work on NAMs, what is being done so that multiple studies don't need to be done across geographic locations? Is there any combined work that's being done to ensure consistency across regulatory authorities?
Yes, absolutely. I mean, the work that is going on with updating the guidance document 280 is being done hand in hand with a big team and with the consensus of ECHA, they're being kept up to date. So we know that the European Chemicals Agency are trying to, improve their confidence with it as well. So that's ongoing. It's a very good question, isn't it? Because the OECD test guidelines are seen as you only need to do one test once, that data will stand across every jurisdiction. That comes down to, I think, the individual regulatory authorities themselves. In terms of having confidence in somebody else's work and expertise, I mean, we do talk to each other an awful lot, the regulators, we really do. So I think there's still a long way to go. But hopefully with the tools such as the IATA, we are actually able to, because everything's set out so transparently, we can at least justify where we've got to with that decision, but also be able to share it, because we do share and we're very transparent with each other when we are making discussions and conclusions and decisions. Oh, sorry, Wesley, that felt like such a non-answer because it's very, very difficult to answer that question.
No, thank you. I think that was a good answer. So thank you.
Yep. Thanks. Then let's move on to the next question. Technical question. How relevant do you think is the composition of sediments regarding bioaccumulation? How do you integrate this using these methods?
The composition of sediments?
Yes. Maybe how they affect bioaccumulation.
Yes. So, yes, it's as a regulator, I would say a sediment-containing bioaccumulation study is one of the most difficult ones to interpret, with the uncertainty coming from absorption to the solid matter itself, also ingestion, and then if you're talking about how do you deporate a sediment dweller. At the moment, for us in REACH, it's not a standard. It is not something we would do. I know it's actually used, I think it's the pesticide industry use it, but it is actually still a really important tool. It's an important test guideline. I mean, both the lumbriculus and the even the earthworm ones as well. However, trying to read those data back to a BCF, that's not very straightforward and it's not very easy. I mean, there are a few publications out there from many, many years ago, but again, they were built on an understanding from the neutral lipophilics. Whether that's changed, whether those metrics still hold today, it was all done from modelling back in the day. So I can put a reference online for you. However, it was not picked up in the test guidance that was carried forward or agreed in Europe when the test guidelines were being issued. So, but it's still worth knowing about it.
Question for Pippa. I want to ask if your study includes ionic and or surfactant type of substances.
Yes, it does. This has been our they're wonderful, the surfactants and ionic, especially ones that have both properties. Yes, it's been a big part of when we've been selecting reference substances. What's the ideal? What's the most appropriate? Can we do it with only one functional group to allow us to baseline? So yes, we are accounting for that.
Yeah, and I'll just add that for related to that, I know there's a lot of interest in the ionizables, that one of the things that's coming out of this IVE work is to ensure that those models for the neutrals and ionizables are sort of harmonized across as well. So there's a lot of increased focus on those.
Okay, thank you. And I'll also try to pull together some questions regarding, do you consider that an in silico study is sufficient for performing a risk assessment for bioaccumulation? And what is the regulatory take actually on the NAMs for a bioaccumulation assessment?
Do you want to take that one, Pippa?
Thank you. In silico for bioaccumulation, very, very useful for screening. And that's where at the moment we're sitting, we're trying to, and that's why it's so important, because it's more important almost to screen out sometimes than it is to screen in, but they have to work. In terms, so, but it would still be used as a line of evidence in the final discussion. That's a really important thing to remember. Where we can't use in silico, we'll often use read across, and that can also involve doing read across using the B models. So you'd actually run a substance that's got the data, the empirical data available, you put it in the in silico. If you can justify that it is similar enough that it's going to behave that way, yes, that could probably be done. But it is substance by substance, but also, you know, jurisdictionally dependent.
Thank you.
Another question, how do the BET and OPERA models improve upon EpiSuite, and what specific benefits do they offer for making more reliable predictions than EpiSuite?
Oh, that's a good question. EpiSuite and OPERA work slightly differently. And it also depends on the unique module within the, so we have the BCF BAF and the BCF WIN within EpiSuite. OPERA can pull on those and pull on the validation data and the, what do you call it? So your testing data and your validation data, which are the same, but they're actually quite old. They're available online actually at using EpiSuite. OPERA goes slightly further. I think it uses the same base data, has slightly different algorithms in it. But the BET tool, which is in EasySuite, not EpiSuite, it's in EasySuite, is by far much stronger at the moment because of the way it's been updated and the different algorithms have been incorporated.
Yeah, I'll just add to that. I think all of these models have utility, of course. I think the ones that are in EasySuite, both in the BAT and the BET, they're drawing from different, say, for example, KOW prediction models, right? Like some of the IFS QSARs as well, and also for the biotransformation. So they're pulling from different phys-chem property prediction estimates, as well as, of course, measured information, and allow you to kind of look at sort of overall consensus, even at the phys-chem property aspect. And then, as I mentioned, for that DCHP example, one of the things is that even though there is a biotransformation aspect in EpiSuite, it's limited related to kind of how those are fit based on different fragments. So there's sort of advances that have been happening within the biotransformation space that's integrated as well. And I think a lot of this, it's important to note that over the last, decade at least, if not more, there's been really significant advances in what we're able to measure and the models that have been developed, and it's just a matter of sort of keeping up with the Joneses, so to speak, with those. So I think that there's a lot of utility, and that's why I think what we tried to do with the IATA is look across all of those different models to try to understand why one might be more applicable and better for certain chemistries than others, for example.
Thank you so much. This is a great discussion, but for time reasons, we need to wrap the webinar up now. So thank you everyone for your input, questions, and participation. Of course, we are grateful to our speakers for their time and expertise. Please keep an eye out for future webinars in the EcoNAMS webinar series the website is on the slide. We will also be sending out a survey and would greatly appreciate your feedback. As we said in the beginning, we will make the recording available on the PETA Science Consortium International YouTube channel. Enjoy the rest of your day, and bye.